Good morning.
Good morning, sir. Good morning, sir.
Yes. Still, there are. Okay, we have only 17 who have joined today. Fine. So let's begin. So we were talking about the examples as far as the tertiary structure is concerned in case of the fibrous protein as well as the globular protein. So we have already seen in case of the fibrous protein, the structure is very simple because you have the linear type of the structures which could be present, and these linear structures can acquire either the helical structure or the beta sheet structure. So in case of fibrous, to understand the folding of the protein or the overall structure of the protein, it is very simple, but it's very difficult for the globular proteins, because within the globular proteins we know that the polypeptide is folding on itself. It is changing its direction by having some turns, loops, or the bulges. So we will not be able to understand that how the actual folding of this protein might have taken place. Okay, but still, through the different researches what we have, we are trying to see some of the glimpses that how a particular globular protein could be folded. What are the things which could be present within the protein, that is in the interior or the core of the protein, and what are the things which could be present onto the outer side, right? So few of the things which I have already told you. So the common thing when you talk about the fibrous proteins and the globular proteins is that in the globular proteins also we have the alpha helices and the sheets. Okay, but now depending upon the nature of the protein, these alpha helices or the sheets, they could be either present into the interior of the protein or they could be present onto the outer side. Now the basic logic which you people can now understand, that if these helices or the sheets, if they have to be into the core or into the interior of the protein, what should be their nature? Whether it should be hydrophilic or whether it should be hydrophobic. So one thing what we can say that it might be dominated by the hydrophobic residues, so that they can be uh, taken inside the cell to form the core of that particular, not the cell, but taken inside the protein to form the core of that particular. But if it is having some hydrophilic residues, those hydrophilic residues, they are involved in formation of the extensive hydrogen bonding. Okay, and due to that, it gives a neutral nature to the helix or the sheet. And that is how we can imagine that this helices or the sheets, they are present in this particular form in the interior or the core of the protein. Now the second thing comes, that what about those helices or the sheets which are present onto the exterior? Again, in that particular case, the helices or the sheets, they are amphipathic in nature. Means what? Means they are having two sides, where one side is hydrophobic and the other side is the hydrophilic side. So obviously the side which is hydrophobic in nature, it will be facing towards the core, or it could be slightly embedded into the core or the interior of the protein, while the remaining part, it could be present onto the exterior or the outer side of that particular protein. Right? So this is how the helices or the sheets, they are present into the globular proteins, or they are also dominating the structure of the globular proteins. So in that case, I have shown you some images just to understand that if a helix is present into the core, or if it is present in somewhere in boundary, or if it is completely exposed to the solvent, that it may have a complete hydrophilic amino acid residues which are present, which is forming that particular structure of the protein. Right? So to move further, And to understand much more things about these globular proteins, I'm presenting the screen so that we can start with the next part now. Just let me know whether you can see the screen.
Yes, sir.
Okay.
Yes,
Fine.
sir.
Okay, so this is what is the examples which I have shown you, that this is a protein which is amphipathic in nature, that is half part is embedded and half part is exposed to the solvent. The next example what we have seen was that of a protein which is completely embedded into the interior. So it is dominated by the hydrophobic residues, and you have this one more example which I shown that it is exposed to the solvent. Okay, so you can imagine that the residues which are present onto these helices, they are hydrophilic in nature, and that is what is the sequences which has been shown. Now, based on this information, we have seen that these proteins, they can be classified on the basis of what is the core which is made up of and what is the backbone. So I told you core is what? It is the interior or the region which is trapped into two different either helices or the sheets, and the backbone is nothing but the polypeptide backbone. So based on these particular two aspects, we can classify the globular proteins. So yeah, this also we have seen. So I told you that majority of the proteins, or more than half of the proteins which are present into the cell, they are of this category, that is two layers of backbone which are separated by one hydrophobic core. Fine. So this is what the backbone can be seen as, because we are talking about the main chain of the polypeptide, and within these two backbones you can see that the core region which is made up of the hydrophobic residues or the R groups of that particular polypeptide chain. So majority of the protein, they could be of this category. Then you have one third of the proteins, of the total proteins I'm talking about. So they could be of this category, that three backbone and two cores. Okay, and there are few proteins which could be having either four backbone, three cores, five layers, four core, or maybe six layers, five core, like this. Now you can imagine, but these are very few examples, or one or two examples which could be of that category. So from this, as I told you, that we can imagine that nature has not selected too much of complication into the protein folding. Okay, so there are very simple structures which are present, so that these proteins, they can be existing or they can carry out some specific functions, right? So these are few of the images which I have already shown you. Yeah. So depending upon, again, depending upon that whether that protein is being made up of the helices or the sheets, we can then classify. So it has been classified into these four broad groups. Okay, like in fibrous protein, I told you about the keratin. So you can classify it as an alpha keratin or the beta keratin. The same way, the globular protein, they can also be classified based upon which type of secondary structure is present into that protein. So in that, the first one is which is made up of the helices. Now these helices, they could be linear in nature. Sorry, obviously they are linear in nature, but they could be directed in one particular direction. But uh, as we are talking about the globular protein, means there is going to be turns, there is going to be loops, so that the direction is being changing. So here you can now say that the even if the helices are present, but you may have the parallel or the anti-parallel type of the helices, right? So this is just with respect to the change into the direction we are talking about this helices. Otherwise, helix structure you do not have as parallel or anti-parallel like what we have in case of the beta sheets, but it is just an arrangement, that they are arranged either in parallel fashion or into the anti-parallel fashion. So here in this case, we are talking about the anti-parallel fashion. So some of the examples which I have shown you here, see, so it consists of the helices. So you can see the bundles of these helices. Here also you can see there is a bundles of helices, but in addition to that helices, you may have small amount of the sheets which could be present here. Then you have this myoherbethrine, you have the heteroglobulin, etc., etc. So these are for some of the examples where you can say that these are the proteins having the helices present into them. Now these helices which are shown here, just to show you, I don't know whether you'll be able to see the arrow or not. So if you are looking for this, that the polypeptide is moving like this, so now it has folded, it has bended. So you can say that if this is from N to C, now here this side it becomes N to C. So in this case, these two helices they become anti-parallel. Okay, again it is changing the direction. So again these two they become the anti-parallel to each other. Fine. So that way you can say that these helices can also be arranged in an anti-parallel fashion. The next category could be a combination of this. That is, you may have the sorry, not combination, but you it is dominated by the beta sheets. So here we see some examples of the proteins which are having the beta sheets. So as I told you, in case of the helix, you have the two sides. The same way you can imagine that for the beta sheets also you have the two sides, where one side can be hydrophilic, the other side can be the hydrophobic, right? The structures, the typical structures in case of those proteins which are having these beta sheets, they could some specific structures are like that. You may have an 8-stranded parallel beta barrel. Okay, means a barrel. I think all of you know what is barrel. You have a hollow structure, a circular hollow structure, which looks like a barrel where you can store the water. Okay, so you have that barrel which is formed by these beta strands. Okay, so there are 8 strands, and these all these sheets they are parallel. Okay, the example is triose phosphate isomerase and pyruvate kinase. So I'll show you the image, and then you may imagine what could be present. So you can see here, in case of the triose phosphate isomerase, so these sheets which are shown, all these sheets they are parallel in nature, and they are forming this circular structure which looks like a barrel. Fine. Now within this triose phosphate, along with the sheets, you also have the helices. But now here we are talking about a category where which are dominated by the beta sheets. So this could be one of the example what we can see. Okay, but although it also contains the helices. So you have an interior or the inner layer of the beta sheets, and exactly covering that particular interior layer, you have the helical layer which is forming that particular protein. And you have the double bound parallel beta sheet. Okay, means maybe you can imagine here now that the middle side could be made up of the beta sheets which are parallel in nature, and both the sides of that particular sheets you may have the helices. So that is why we call it as a mixed also. So you have the parallel or the mixed beta sheet type of these proteins. So just look at the images and you get an idea. See, in the exam, you are not supposed to draw these images or something like that, but this is for your understanding and to show you that how this polypeptide is folded and how it is forming the different shapes and sizes of the protein. Now look here, the example which is shown is a pyruvate kinase. Now here you can see that these sheets, they are all parallel in nature. Okay, and on the both side of this, you can see that the helices are present. Then just look at this hexokinase domain. Now, can anyone tell me, looking at this structure, what type of sheets can you see here? Whether they are parallel or anti-parallel? I think I'm audible.
Yes, sir.
So tell me, what looking at the directions, what you can say here, all the arrowheads are on one side. So you can say that they are parallel. But in this case, if you see, if you compare these two strands, their arrowhead directions are different. So this is having the anti-parallel beta sheets. On both the sides of these beta sheets, you can see that the helices are present. Now see here, in case of flavodoxin, what type of sheets are these?
Parallel.
Parallel.
So these are the parallel, and both the sides you can say that the helices are present. What about this?
Anti-parallel.
Yeah. So it's a mix here again. It could be some are parallel, some are anti-parallel, right? So like these structures can be seen in case into this particular category. Now the third category which you may come across, that is the specifically which are made up of the anti-parallel beta sheet proteins. So in this case, हे बघा आता अँटी पॅरलल आहे म्हणजे काय होईल, जस्ट इमॅजिन means there is a bending due to which the direction has changed. So both the sides, I don't know whether to how to show it. Maybe I'll show it onto this. I have already shown in the last lectures, but still I will show it into the whiteboard. Let me start it. Yeah. Say suppose this is a sheet, one polypeptide which has formed a sheet. Now when this sheet is formed, this side is hydrophobic. So I'll write it as hydrophobic. HB I will write. So just for understanding, I am putting this, and here it should be hydrophilic side. Fine. So I write it as an HP, hydrophilic, and this is hydrophobic. Now this polypeptide is being formed, but now there is a turn. So like this, a turn has taken place. Okay. Now due to this, what has happened? It has become anti-parallel because the direction has changed. But even though if the direction is changed, what has happened due to that? So you have this hydrophobic region which is onto this outer side. So here also you get this side which is hydrophobic in nature. Okay. While the inner side, it remains the hydrophobic. I think you got this particular idea what I want to show it here. Right. So this way, whenever an anti-parallel beta sheet is formed, in that case, you have both the sides which could be either hydrophobic or which could be either hydrophilic, depending upon what type of amino acid residues is making up that particular polypeptide. Now, in this case, I shown you that these are the hydrophobic residue R groups which are pointed outside. It could be the hydrophilic residues which could be pointed outside. Right. So that way you can imagine, which will help you to understand about this particular type of the protein called as an anti-parallel beta sheet proteins. Okay, so as I told you, they arrange the hydrophobic residues on just one side of the sheet with one side exposed to the solvent. Here in this case, it is two layered hydrophobic faces. Okay, in between you have the hydrophilics. That is called a juxtapose, means which is entrapped between these two hydrophobic faces. Now here also the domains are formed by these beta sheets, which could form a shape which is similar to that of the barrel. Okay, and in this case also they are basically what you can say stabilized by the few hydrogen bonds, but maximum stabilization is by the hydrophobic interactions. Now this beta barrel structure which are formed, they could be either parallel or they could be anti-parallel, right? So example, you can quote it could be the soybean trypsin inhibitor, then you have the concavalin A, then gamma crystalline, or glyceraldehyde 3-phosphate dehydrogenase, or glutathione reductase. So these are some of the examples which can be quoted here. Fine. I'll show you the images. From that you will come to know. Now basically these these proteins which are dominated by the beta sheets, they form the Greek key topology. Now this Greek key topology means could be that those proteins, they look like the pyramid, or they look like the utensils which were used by the Greek people, like the vase, Okay, where we keep the flowers, the flower pot ज्याला आपण साध्या शब्दात म्हणतो. So they may look like that particular structure. So that particular type of the folding is called as a Greek key topology. Okay, so I'll show you some beautiful images whereby some specific structures are formed into these proteins. Yeah, now the Greek key topology. So these three examples which are shown, they are of this particular class or the family that is called as the Greek key topology. Okay, yeah. So this see some of the more examples which I have shown here. So all these proteins, they are having the Greek key topology. Now I'll go back to the whiteboard. Now, when you're talking about the folding of the protein, if the protein has to be folded, so you may say that, okay, so it is folding like this only. Okay, just I think you get the point from this. Okay, so it should fold like this, but the folding is not in a sequence manner like this, or it is not sequential. It may change. Maybe, or if I tell you that you draw a star, जर तुम्हाला स्टार काढायला सांगितलं तर तुम्ही कसा काढाल? So if I want to have it in a simple way, I may draw it like this, that this is how the star it is looking like. Okay, but then it is not giving that feel of a star. If I have to draw proper structure, I could change the way this folding has taken place here. So I may draw it like this. Okay, so here in this case, when the folding is taking place, this folding is not sequential. Not necessary that the regions, they are in a sequence manner. You may have the region which is far away, which is closer to each other, and those which are closer to each other, they may be far away. Okay, now when I have joined these 2 points, what has happened? The N terminal and C terminal has come closer to each other, and see the structure what I have got here. So this is a sequential structure, and this is a random folding, you can say. Now if you look at this, I will say that, okay, this is a very good structure of a star as compared to this particular thing. Fine. So it's not necessary that when the folding is taking place, in that folding it is a sequential type of a folding. Okay, so from these examples which I'm trying to show here, you will be able to understand. We'll go back here. Yeah, just 1 minute. Yeah. So see, the polypeptide is starting from this side. So what you should have imagined that, okay, this region, the fourth line what you see here, it should have been here. But what has happened, that folding has changed. So you can see here that it is going this way. So these regions which were closer to each other, they have moved far apart. And ha jo region लांब होता, it has come closer. Now this has come more closer to this region. So you can see that it has followed this particular path. And due to this typical type of a fold, you can say that it is forming the Greek key topology type of the proteins. Okay, so which is giving a specific size and shape to that particular protein. So few more examples or images which I'll try to show you. Okay, so see here, this is the simplest topology. आलं लक्षात? हे simplest, जसं मी तुम्हाला सांगितलं की it is in a sequential manner. की हा इथं फोल्ड झाला, परत फोल्ड होऊन असं. So this is a very simple type. Okay, so it forms the hairpin type of these structures. Now within the Greek key topology, what is happening? See here, if I'm changing the sequence, जर मी इथं नंबर दिले बघा, आता हा 1 2 3 4 5 6, if I give it a sequential numbering, so it is in a sequence 1 2 3 4 5 6. But here in this case, or in case of an example of plastocyanin, which is forming a Greek key topology type of a protein, so see the sequence has changed. So in the starting you have the 2, then 1, then the third region, then the sixth region, then the fifth, fourth, seventh, eighth. म्हणजे बघा, तो फोल्ड होतोय, तो इकडे गेलाय, मग again it has folded here, then this part it has moved far away, and then this region it has come closer, again it has come closer, and again it has moved far. Fine, like something like this, the folding is taking place, and due to that you are getting this shape or the size of the protein which is shown into the image. The same way here you can, the example which I shown you about the gamma crystalline, in that also if you see the sequence is far more different as compared to the plastocyanin. Just compare it with the plastocyanin, this sequence, that is 3 2 1 4 7 6 5 8, and due to that you have a in all a different type of a shape to this particular protein. Okay, so something like this. So this is what is the Greek key topology. It looks like the vase or the pyramid which I told you in case of the Greek. Okay, then some more topologies which I'll try to show here. It is called as a jelly roll topology. See the sequence, 1 2, the 3 it has moved very far apart. हे बघा 3 इकडे आला, लगेच 4th जो region आहे, it has tried to come closer here again, then the 5th. See the way it has been folded. Okay, and then it is going outside like this. So it is forming a jelly roll. Jelly roll म्हणजे चॉकलेट. इमॅजिन करा, चॉकलेट कसं असतं, दोन्ही साइड मध्ये ना फोल्ड असतो आणि मध्ये तुमचं ऍक्च्युअल चॉकलेट असतो. सो इट लुक्स समथिंग लाईक दॅट, अँड दॅट इज वाय इट इज कॉल्ड एन जेली रोल टोपोलॉजी. Right? Now this folding you look at, this is in a neuraminidase. Okay, so it looks like a propeller. So as it is made up of the beta sheets, so it is called as a beta propeller. Right? So there are many beta sheets which are folded in a sequential manner, and then you get such kind of beta propeller structure. So the protein may look something like this, which is shown here. Yeah, these are some of the different type of the foldings in case of the Greek key topology. Okay, so for the different protein you may have the different type of the structures which could be obtained, and these structures could be the variation into the sequence of this folding. Yeah, the two more topologies what you can see here, that is why the first one is an horseshoe. Horseshoe, I think all of you know, there is a nail, some iron thing which is being implanted onto the legs of the horse. That is why you hear that noise, knock knock knock. So that is an horseshoe upon the. So this it looks like an horseshoe structure, and that is why it is called as an horseshoe type of an topology. Now this is a very beautiful type of folding that is called as an Rossmann fold. So you can say it is a continuation of this horseshoe, that you are having that horseshoe which is continuated many a times, and that type of a fold is called as an Rossmann fold. If I show you the image, it looks very beautiful. See here. Okay, so it looks something like this. Means the inner side of these particular proteins, they are made up of the sheets, and all these sheets they are parallel, and covering these sheets you have the helices. Right? So this particular type of a fold is called as an Rossmann fold. Now here you can see here many other types of the folds which are of the category that is the Rossmann fold. So they are what you can say categorized or classified as Rossmann fold 1, 2, 3, 4, 5. Okay, so simply the sequences change and you get a different type of a protein. Right? हे फक्त तुम्हाला समजण्यासाठी किंवा तुमच्या माहितीसाठी मी हे सांगतोय. यू आर नॉट सपोज टू ड्रॉ दिस डायग्राम, बट फ्रॉम दिस you should be able to imagine that this such kind of variation into the sequence of the folding is going to give you the different shapes and size of the proteins. See some more examples what you can see here. Just look at the images, how beautiful these proteins they look like, the structures, the way they are folded. Yeah, one more sandwich. I think all of you had the breakfast, so don't get tempted. So sandwich means what? That you have the sequence of the beta sheets, okay, and that whole sequence is being layered onto the another sequence, so which looks like a sandwich. So you have two different layers which are put on each one on each other, so you get an appearance like that of an sandwich. Then you have this barrel like structure. See here how beautiful this barrel like structure it looks like. so that they are called as the beta barrels. These are some of the structures of the beta barrels. Okay, although I'm not talking about the examples, but just look at the shapes and size, which will give you the idea. So that was about the third category, which is dominated by the anti-parallel beta sheets. Now we'll move to the fourth category, fourth type of the classification, where along with these helices or the sheets, you have some metal which is being embedded into it, or those proteins in which the disulfide bonds are more prominent or which are present, which is giving the further stability to that particular protein. Fine. So here these proteins which are of this category, that is containing a metal or which are rich in disulfide bonds, they are these smaller proteins, maybe which could be made up of less than 100 residues, but they are influenced by the presence of the metals or the disulfide bonds within that particular structures. Okay, the example could be your insulin, phospholipase A2, crambin. Then there is a protein called as ferredoxine. You have another protein called as an high potential iron protein. So these are all the proteins which are responsible for sequestering the iron, means they can accommodate the iron in their structure. So whenever required by the cell, these iron they are released, and they can be provided to the cells. Okay, or you might have heard about the calcium binding protein, one example which I have shown you. So that particular type of the proteins, they come under this category. Fine. So they may have the similar structures what we have seen in the earlier cases. Fine, but there are some distortions which could be observed here. So these are some of the examples what you can see here, like insulin, crambin, phospholipase A2. Then these are the ferredoxine proteins. Now within these proteins, I don't know whether you can see here, see some lightning sign is shown in the yellow color. These small lightning signs what you see, these are nothing but the disulfide bonds which are further stabilizing that particular protein, and that shape is being acquired by the protein, right? So this is about the fourth category, where you can classify the proteins based on their uh, based upon the secondary structures which are present onto that particular proteins. Yeah. Now next, in case of the globular protein, which I told you that it could be dominated by the helices or the sheets. But in addition to these helices and the sheets, the globular protein, they have some other regions also, which could be involved into the formation of the turns, loops, or the bulges. Okay, so what are those particular regions? So in that, one of the region which you can say that it is an ordered and a non-repetitive structure, which is responsible for that particular type of the turns. So what are these regions? So these are the segments of the polypeptide chain which cannot be classified into having some specific secondary structures. So it is simple a coil, or it is a random coil, what you can call it as, because it does not have any alpha helical or an beta sheet type of the structure. So these are neither coiled nor random, but they are highly organized and they are stable, and they are also strongly influenced by the side chain interactions. I'll show you the images and that will give you an idea. So these structures, they are very interesting type of the structures, because these are the structures where you can go for the protein engineering. Okay, so scientists are trying to focus onto these particular regions, although we are not able to understand the actual function of this particular region, but still the research is going on, and we are trying to understand what could be the specific region for it. It has been found that these structures, they could be the structures which are present into the enzyme active site, or the site where the antigen antibody interaction is taking place, or it could be the site where a particular protein is interacting with the receptors onto the cell, etc., etc. So they have some important function at those particular active sides. So example could be your myoglobin, which we know which is responsible for oxygen transportation. Then pancreatic trypsin inhibitor, it will go and bind to some specific site of the protein. It will inhibit the activity of that protein. So such kind of proteins, they are having this order or the non-repetitive structures. So see here the image. One example which I have shown here, it consists of a helix, it consists of the sheets. Now if you see that within the helix or within the sheet, it is stabilized by the hydrogen bond. But then these ordered or the non-repetitive structures which are present, they have their R groups which are free. Okay, so it might be containing hydroxyl group, it might contain a carboxyl group or an amino group, or it could be a sulfur group. So those free functional groups, they can form the hydrogen bonds with the secondary structures which are. So see these orange color lines, orange color, these regions which are shown, they are nothing but these non-repetitive structures, ordered and non-repetitive structures. And the residues which are present into these regions, they can form the hydrogen bond with the helices or the sheets which are present into the protein. So see here, this aspargine, aspargine's structure, it consists of around 2 to 3 amino groups. So all those amino groups, they are involved in the formation of this hydrogen bond. Okay, so this interaction is also giving or it is also contributing toward the specific shape and size of that particular protein, right? So this is one of the type of the structure which could be present into the globular protein. In addition to this, you have some regions which are the disordered segments and which are flexible. Okay, means the sequence is not what you can say fixed. Okay, so it could be a random. If you try to compare it between 100 proteins, in all the proteins you may get that the sequence is very different from each other. While in the previous one which I told you, that is the ordered structure, there the sequence is fixed. If that region has to be present into a protein, it will be fixed. But here in this case, it is disordered, but it is giving the flexibility to that particular protein. Okay, so genuinely disordered segments. These segments, although they cannot be easily seen or studied in using the X-ray crystallographic studies, but these segments are the one which actually play an important role during the conformational changes which are taking place into the protein. Right? So that movement within the protein is being brought about by this flexible or the disorder segment of that protein. Right? So it is common for long charged side chains on the surface of the proteins. So there are many examples which you can quote here. So it could be the presence of the lysine. Lysines are R groups that it is having a very long chain which is containing amino groups into it. So such kind of residues are present into that disorder segments. Fine. So in addition to the helices and the sheets, you have this ordered segment. Okay, the ordered segment which is a non-repetitive structure, and you have this disordered segment which is giving the flexibility to the protein. Now, knowing this, I just want to ask you a question. What I have told you that a given globular protein is having the core region, the interior or and the outer region. Now this core region, it is made up of the helices, the sheets. It could be this different type of the segments which I told you. It could be the disordered segments or the ordered segments. Now is it that this core of the protein, it is compactly packed? What do you say? Whether it should be compactly packed or it should not be compactly packed? Any views which if I can hear from you people? Yes. I think I'm audible.
Yes, sir.
Yeah. Can you tell me? एखादं हे बघा, तुम्हाला पॉलिपेप्टाइड दिलंय, तुम्हाला सांगितलं याला फोल्ड करायचंय. फोल्ड तुम्ही मग जे काही कॅरेक्टरिस्टिक त्याप्रमाणे तुम्ही फोल्ड केलं, मग तुमचा इंटिरियर जो आहे तो एकदम असा कॉम्पॅक्टली पॅक पाहिजे का नाही पाहिजे? व्हॉट डू यू से? जस्ट युअर व्ह्यूज, आय वॉन्ट युअर व्ह्यूज. जस्ट से ऍटलीस्ट फर्स्ट येस ऑर नो तर म्हणा, मग त्याच्यावर आपण डिस्कशन करू ना.
सर कॉम्पॅक्टली पॅक.
कॉम्पॅक्टली पॅक पाहिजे. ओके. एनी अदर? वाय डू यू वॉन्ट इट टू बी कॉम्पॅक्टली पॅक? थोडस रिझनिंग देऊ आपण त्याला. का तुला वाटतंय की ते कॉम्पॅक्टली पॅक पाहिजे, किंवा ज्याला कोणाला वाटत असेल की तो कॉम्पॅक्टली पॅक नको पाहिजे, तर का पाहिजे, का नको पाहिजे?
असं प्रॉपर फोल्डिंग होणार नाही ना.
ओके, प्रॉपर फोल्डिंग होणार नाही. अजून काय कोणाला सांगायचंय? येस. ओके. आता ही जर म्हटली, ज्यांनी कोणी आता उत्तर दिलं मला माहित नाही कोणी दिलं. सो इफ यू से दॅट इट इज कॉम्पॅक्टली पॅक, मी त्याला पूर्ण 100% पॅकिंग म्हटलंय, पूर्ण पॅक केलंय. But if I want some changes to happen into that protein, if a substrate is trying to enter into the protein, can it enter? तुम्ही एकदम त्याला रिजिड बनवून ठेवलेलं आहे. So can you imagine that there is some conformational change which can take place into that rigid structure? No. Okay. So many examples what we have in front of us, that these proteins or the enzymes, they are undergoing some specific type of the conformational changes. And if you have to have some specific conformational changes into the protein, your protein should not be a rigid protein. It should not be compactly packed. There has to be some amount of flexibility available into that particular protein. So studying the different type of the proteins, where the conformational changes is going to take, what they found, that the compaction into to those protein is up to 77%. So the remaining, it could be how much? 77% मी जेव्हा इथं म्हणतोय, so or the remaining I think 23% is some or the other kind of the space which is present into the interior or the core of the protein. And this space which is present, it is giving the flexibility to the protein so that it can have many different type of the conformational changes. Okay. So the one example which I have told you about the DNA polymerase. See, in the DNA polymerase, what happened, that there is a cleft. So the cleft, it open up, and within that cleft you have the what you can say, the single stranded DNA which is entering. Now once the DNA it has entered, or it is being trapped into the cleft it closes. So if I want to have these changes of the cleft, okay, so for that I should have some space which can accommodate that particular movement, and that is why there is no 100% compaction which is found into the proteins. Fine. So there is around 75 to 77% of the compaction, while the remaining amount is a space which is present, and that allows the that allows the different type of the changes which can take place into the protein. So what kind of movements can you imagine which could be happening into the protein itself? So there are three types of the motions or fluctuations which can take place into the protein. Okay. So the first could be your atomic vibration. Now see, you have a protein. Protein is made up of different type of molecules, and they are the amino acids. These amino acids, they are made up of the elements, and elements, they have the atoms. So you can imagine that if there is a change which has to take place into the protein, or if there is a motion or a movement which has to take into the protein, it could be at an atomic level. So you may have the atomic vibrations which could take place. Okay. Then you may have that some region of that protein is moving. जसं मी तुम्हाला सांगितलं की डोमेन्स, आता किंवा एक एक्झाम्पल आता क्लेफ्टचं सांगितलं. सो जस्ट इमॅजिन दॅट दिस टू क्लेफ्ट is made up of two domains. Means these two domains, they are moving away from each other, again they are coming towards each other. So you have those collective motions. So it could be collective motion of some 2 to 3 atoms, or it could be 2 to 3 molecules, or it could be your complete domain which is moving. So you have that kind of collective motions which are taking place, right? And for both this type of the motions, that is the atomic or the collective motions, you simply require the kinetic energy which is present within the molecule when the interaction is going to take place. In addition to this two type of the motions, you have the third type of motion which could be there, that is called as an triggered conformational changes. Okay, means what? The example I can take about the glycogen synthesis, where I told you that simply the phosphorylation and dephosphorylation is changing the nature of the protein, means you are triggering. So the moment I phosphorylate, what is happening? There is a conformational change which is taking place, due to which the active site it has opened. Okay, and if I remove the phosphate, again the conformational change, the active site is closed, right? So to have this different type of the motions or the fluctuations into the protein, the compaction should not be 100%, right? So there is some or the other amount of space which is present, which is giving the chance of having such kind of fluctuations or the motions into the proteins, right? So I think you got an idea that what is actually happening into a globular protein. So globular protein in all सगळं आपण एकदम नाही सांगू शकत, but you can say that it is dominated by the alpha helices or the sheets or the combinations, right? In addition to these secondary structures, you have some other regions which are not having any helical or a sheet structure. Okay, but they could be ordered, they could be disordered, but they are giving some specific characteristic to your protein, etc., etc., right? So with all these things what I have told you, you should be able to understand that how a globular protein is being formed or how it is functioning. So after knowing this, the question comes that how do proteins know that how they have to fold? And that is what we are going to discuss in the next lecture, that how actually the proteins they fold. Okay, so I'll stop here. Any questions up to this point which I have discussed? Yeah, any kind of question or any slide which you have not understood, any point which you have not understood, please raise some questions. That is what I am requesting every time. Meanwhile, you can put your roll numbers in the chat box. Snehal, are you there? Snehal
Yes,
Chau.
sir.
Write
Yes,
down
sir.
the numbers and send me.
Okay, sir.
Yeah. Any questions?
No, sir.
ओके. बरं, आता हॉस्टेलला सगळे आलेत का?
Not yet, sir.
What's the problem? Why they are not coming?
Don't know, sir. Might be they will come